Hi everyone, it's Dr. Alia again for Bio 3003 Plant Structure and Function. We're going to continue with plant growth in roots. The growth of roots in terms of its length is concentrated in the root tip area where there are three zones of cells with successive stages of primary growth. So we have the zone of cell division, zone of elongation, and zone of maturation. For zone of cell division, it consists of the apical meristem and primary meristems, its derivatives, and also a quiescent center. So near the middle is the quiescent center there, and let me show you here, quiescent center there. Yes, and the cells here divide more slowly than other meristematic cells. These cells are relatively resistant to damage from radiation and toxic chemicals. So they're quite special because they act as a reserve that can restore the meristem if it becomes damaged. Next, we have the zone of elongation. So the zone of elongation blends into the zone of cell division where the cells elongate, sometimes to more than 10 times its original length. So this elongation of cells is mainly responsible for pushing the root tip, including the meristem, into the soil. So the meristem sustains the growth by continuously adding cells to the youngest end of the zone of elongation. Then we have the zone of maturation. So here cells begin to specialize in terms of their structure and function. In this root region, the three tissue systems produced by the primary growth complete their differentiation and their cells becoming functionally mature. The three primary meristems give rise to the three primary tissues of roots. This is a very nice image here where you can see that everything is color-coded, and based on the colors you can see where certain cells come from. And if you look here, you can see that they all come from the quiescent center, that's the QC there. So first we have the epidermis that develops from the dermal tissues, so that we call protoderm. And then we have the vascular tissue that produces the stele, the pericycle, the pith, the xylem, the phloem, so that's the procambium, and the ground tissue that produces the endodermis and the cortex. Let's have a closer look at the three primary meristems. So you can see here it's color-coded. Let's start with the protoderm, which is in blue. So the protoderm is the outermost primary meristem that produces the single cell layer of the epidermis. So water and minerals absorbed by the plant must enter through this epidermis. And there's also the presence of root hairs that enhance the absorption of water by greatly increasing the surface area. Next, we have the procambium. So the procambium gives rise to the stele, which in roots is the central cylinder of vascular tissue. So vascular tissue, so where the xylem and the phloem develop. And the ground tissue between the protoderm and the procambium give rise to the ground tissue system. This is where mostly parenchymal cells between the stele and the epidermis function to store food and are active in the uptake of minerals that enter the root with the soil solution. And one thing to note, I don't think there's it's labeled here. There is an innermost layer of the cortex, the endodermis. Oh, terrible handwriting, sorry. It's a cylinder, one cell thick, that forms a boundary between the cortex and the stele. So from the primary growth of the roots, they will eventually develop into the mature structures according to their plant types. So when I say plant types here, meaning the dicot and the monocots. So the dicots and monocots are plant types, and I hope you remember about them. So there's different structures between dicots and monocots. In monocots, the xylem and the phloem tissue bundles are arranged in a circular fashion around the central pith. So the central pith consists of the ground tissue parenchyma. And then we have a layer of pericycle that encircle this vascular bundle, and outer to that will be the endodermis, cortex, and epidermis. Compared to the monocots, the arrangement of xylem and phloem is different in dicots. The xylem in dicots is all located in the middle of the dicot root, and bundles of phloem are arranged around it. And so it's separated by the vascular cambium. So in dicots, they differentiate into secondary growth xylem and phloem, which increases the girth of the dicot roots and stem. Monocots do not have a vascular cambium. So since dicots don't have a central pith area, the parenchyma serves as connective tissue in the region where dicots roots vascular structures are found. Since we are talking about monocots and dicots, I think let's just go have a refresher and remember the differences between monocots and dicots. So first of all, in terms of its cotyledon, monocots have one cotyledon within their seeds, dicots have two. In terms of their roots, monocots have typically fibrous or adventitious roots, and dicots have a main tap root with lateral branches. In terms of their roots, in terms of their vascular tissue, stem vascularization, the vascular bundles in monocots are scattered, whereas they form a ring structure in dicots. In terms of the leaf veins, monocots show parallel venation, whereas dicots display reticulated or net-like veins. And lastly, in terms of the floral organs, so for monocots, they have flower parts in multiples of 3, while dicots are in multiples of 4 or 5 in terms of their flowers. So that's all for today's for this video. I'll see you next time. Bye.